Hello and welcome to this session. Today we will explore how enzymes drive essential reactions in the body. We will examine their structure and function and understand how these biological catalysts speed up chemical processes that are vital for sustaining life. Enzymes are primarily globular proteins composed of long chains of amino acids. Their function is intrinsically linked to their structure. The primary structure is a specific sequence of amino acids linked by peptide bonds. This sequence dictates the folding into secondary structures like alpha helices and beta sheets. These folds are stabilized by hydrogen bonds. The tertiary structure is the enzyme's three-dimensional shape formed by interactions between R groups. These include hydrogen bonds, ionic bonds, disulfide bridges, and hydrophobic interactions. The active site is a specific region formed within this structure. Some enzymes have a quaternary structure consisting of multiple polypeptide chains. These chains work together to form a functional enzyme. The active site is a small, uniquely shaped region where the substrate binds. It is highly specific, complementary in shape to the substrate. Its chemical properties are suited to bind specific molecules. This specificity ensures that enzymes catalyze only one type of reaction. Enzyme-substrate interaction is crucial for enzyme function. Enzymes speed up reactions by lowering the activation energy needed to start them. They achieve this by bringing substrates together and orienting molecules correctly. Enzymes strain bonds in substrates and provide a favorable microenvironment. The enzyme-substrate complex forms, then converts to product, releasing the enzyme unchanged. This allows enzymes to be reused in subsequent reactions. The lock-and-key model suggests the active site is rigid and complementary to the substrate. The substrate fits exactly like a key in a lock. The induced fit model proposes that the active site is flexible. Binding induces a shape change in the enzyme for better substrate fit. This model better explains enzyme functionality. Temperature affects enzyme activity by increasing molecular movement and collision frequency. High temperatures can denature enzymes, altering the active site. Each enzyme has an optimum pH, and deviations can change ionic and hydrogen bonds. Substrate concentration increases reaction rate until saturation. Enzyme concentration provides more active sites, enhancing reaction rate. Enzymes are crucial for digestion, cellular respiration, DNA replication, and metabolism regulation. Without enzymes, biochemical reactions would occur too slowly to sustain life. They allow reactions to occur rapidly under physiological conditions. Enzymes ensure efficient energy use and resource management in cells. Their role is fundamental to biological processes. Enzymes are globular proteins, and their function is determined by their structure. The three-dimensional shape allows substrate binding and reaction catalysis. The active site is critical for enzyme specificity. If the active site changes shape, substrate binding is impaired. Enzyme structure is key to its biological function. Some enzymes require cofactors for proper function. Cofactors can be metal ions, coenzymes, or prosthetic groups. They help stabilize the active site and assist in substrate binding. Cofactors can also participate directly in chemical reactions. Without the correct cofactor, enzyme activity is compromised. Enzymes lower activation energy, the minimum energy needed for a reaction. They do this by aligning substrates and straining bonds. Enzymes create a favorable microenvironment for reactions. This allows reactions to occur rapidly at normal body temperatures. Enzyme specificity ensures controlled metabolic pathways. The lock-and-key hypothesis suggests the active site is rigid. The substrate fits exactly into the active site. Enzyme and substrate are complementary before binding. This model explains enzyme specificity. However, it lacks flexibility in explaining dynamic enzyme behavior. The induced fit theory suggests the active site is flexible. Substrate binding causes the enzyme to change shape slightly. This change improves substrate binding and stabilizes the transition state. The induced fit theory better explains enzyme action. It shows how enzymes reduce activation energy effectively. Catabolic reactions include amylase breaking down starch into maltose. Catalase breaks down hydrogen peroxide into water and oxygen. ATP synthase is involved in energy metabolism. Anabolic reactions involve DNA polymerase synthesizing new DNA strands. These examples illustrate enzyme roles in both breakdown and synthesis processes. Competitive inhibition occurs when inhibitors resemble substrates. They compete for the active site, reducing reaction rate. This can be overcome by increasing substrate concentration. Non-competitive inhibition involves inhibitors binding to an allosteric site. This changes enzyme shape and cannot be overcome by more substrate. Temperature affects enzyme activity by increasing kinetic energy. Higher temperatures increase collision frequency and reaction rate. Beyond the optimum, enzymes denature as bonds break. The active site loses shape, drastically reducing reaction rate. Most human enzymes have an optimum temperature around 37 degrees Celsius. Q10 measures how much reaction rate increases with a 10 degrees Celsius rise. The formula is the rate at higher temperature divided by the rate at lower temperature. For many enzyme reactions, Q10 is approximately 2. This means the rate doubles with every 10-degree increase. Q10 evaluates temperature sensitivity in enzyme studies. Each enzyme has an optimum pH for maximum activity. Changes in pH alter hydrogen and ionic bonds, affecting enzyme shape. This can reduce catalytic activity or cause denaturation. Digestive enzymes have varying optimum pH levels, reflecting adaptation. Extreme pH values can permanently alter enzyme structure. Increasing substrate concentration boosts reaction rate until saturation. At this point, all active sites are occupied, reaching maximum velocity. More enzyme concentration provides more active sites, enhancing rate. Reaction rate is proportional to enzyme concentration if substrate is not limiting. Practical applications involve serial dilutions for experimental investigation. Temperature graphs show a gradual increase to optimum, then a sharp decline. This decline indicates structural damage from denaturation. pH graphs display a bell-shaped curve, peaking at optimum pH. Sharp drops at extreme pH suggest denaturation or altered active site charge. These graphs help interpret enzyme activity patterns. Competitive inhibition reduces reaction rate, but is reversible by more substrate. Non-competitive inhibition permanently lowers maximum catalytic capacity. Temperature increases right up to optimum, then causes denaturation. pH affects bonding and enzyme stability. Together, these factors regulate enzyme efficiency in biochemical reactions. Understanding these principles highlights enzymes' critical role in biological systems. For any queries, send us a WhatsApp message at 074-84361-688, and a team member will be happy to help. Thank you, and we look forward to the next session.